files. I'm not sure about the multiple files option here, but maybe you can give them one by one. It doesn't take a lot of time, just a couple of minutes and it will be done. Is everyone following? Yes, forward and reverse files also you can give them one by one because it doesn't. I'm not sure about Galaxy if it takes both of them together because I was able to select only one file. In Terminal, you can give both of them together. I'm mentioning both the files. So for Terminal, I'll give the list of commands for FastQC. Is one will be any other doubts, please switch on the audio and ask me in case I'm not able to view your messages. So once we download from SRA, we need to convert the file to FastQ files. Command will be in the SRA file ID. Then after you get the read one and read two file, you have to execute FastQ. So in case your one more major important thing here is one second. In case your FastQ file does not is not zipped, is not .gz, you can directly convert it. To zip the files, you can use the command gzip and the file name space file name. So this will convert the files to fastq.gz. This will also consume less memory. These files are compressed files. And once you have compressed them, you can directly use fastqc. fastqc and your files can be given. Either you can first to get the help list of commands, you can type fastqc help. Then you can give fastqc and file one.fastq.gz and file two.fastq.gz, or you can also give all the fastq files together, that is fastqc *.gz. So this will run all the files together. You can give the thread command here. Thread can be anything 4 or 5 or 6. Maximum you can give 6 to 10 based on your computational power. Uh, is everyone following? Do I repeat any particular section? Please let me know. So this was right from accessing the raw data, selecting the type of raw data, and yes. So this was right from how do we identify the raw data required for the analysis, and how do we download it from SRA in Linux system, and how do we convert it into FASTQ files, and how do we zip them, compress them, that is, and how do we use FastQC for the quality calibration using the Linux system and using Galaxy Workbench both. Galaxy is pretty easier in case you are not able to, get in line with the command lines. You can directly upload your files and just give the commands in Galaxy. It will give it to you. I can show you the Galaxy. Uh, sure. So in Galaxy, we can directly go to Galaxy Bioinformatics. So it will give you Galaxy or database directly. The last command. So the last command is fastqc. Minus t is the thread, that is computer threads. So I am giving -6 because my computational power maximum I am giving here. But you can also give 10. It doesn't matter, 10 or how much ever you want. And asterisk means to consider all the zipped files. So this is to simultaneously execute all the fastq files in single command. The second command here, I have specified the file names individually and I have done, which is a lengthy process, but I have just explained it to you. But the last command is a very easy command to give all the files together at once. So whenever you want to select the files together, you just have to give asterisks and the file ending. So my files are ending with .gz. Usually our files will end with .gz. Yeah, Galaxy, coming to the Galaxy part. So this will give us all the tools and uh, list of tools we need. So we have common genomic tools, like you can see on the left hand side. You can get the data, you can upload data, you can manipulate the text files, you can sort and filter. So it enables us to do a lot of NGS data analysis here. It also helps us to do the alignment and mapping annotation. RNA sequencing analysis is done very easily here. When we can do the variant calling, which I will be showing you all tomorrow. It's just that it will take a lot of time, and it will also give us the. Yes, so you can upload your data, or you can first search for the tools you want. Now I need genomic file manipulation, that is FastQ quality control. So I'll select the tool, or I'll just first upload the data. 1 second, yeah. Here I can select multiple files here. To address the previous question, yes, you can upload multiple files. You just have to go on the collection. That is, I'll show again. Once you open the Galaxy work database page, you can go to upload data, and you can go to collection. You can go to collection, and you can go to choose local files, and you can select all the raw datas. _1 _2 dot fastq dot gz are the raw data files. So here I'm just currently giving 4 files, and I'll just give. So once it gets uploaded, I will have to click on start, and it will upload one by one. Then I have to. So it will give me the status, and accordingly it will also. It's taking a while. Okay. Then on the right hand side, whatever processes you are running, it will be displaying its uh, status. So here I had earlier given one raw data, and it is like if there's an error, it will display like there's an error with particular this thing. So in the second one over here, it is giving me. So I'll have to log in over here to access. Yeah. First, I'll have to log in and then I can access the web page. But usually, the previous result which I showed, it should be displaying to you like this HTML. It'll redirect to the HTML page. So once the process is complete, the upload process is complete, you can click on the tabs here and it will redirect you to the HTML page like this, where you can take a judgment about the raw data you have uploaded. And you can also download it, save it, like these are the save buttons. You can get all the details. You can retry it if you want it to run again. So I just give a run again. So to run the tool, I just have to click on run tool and I have to just select again. Any doubts until now? Galaxy will give all the output files after using any packages. It will give basic output files like for fast QCI, we'll require only HTML pages. So it will give us HTML page. The save button, you can directly save it. Output files also, maybe we can download them and yeah, we can access them directly. But usually it will definitely let us save the output. So that's not a worry here. Uh, is it clear until now to everyone? Tomorrow again, I will go through the entire parameters. Yes, so parameters for each tool you can select as per you want. I have just given the default uh, parameters, and I didn't. So here, if I want to select, suppose in any other one. Yes, parameters you can select based on your study, but I have given default parameters. You can first proceed with the default parameters and accordingly adjust it if you are getting a very vague data report. Aishwarya, Aishwarya, please let me know your query. Hello, hello. Okay, okay, that's not a problem. So this was just the data downloading this thing. So what I'll do is I'll give you the list of tools we require. If not, also it's okay. We can access them through Galaxy. Yes. For parameters, you need not worry about it. I mean, it also takes it without giving any parameters for analysis. And so Galaxy takes a lot of time, and Linux tool is much more handy for us, to analyze all the files together, visualize them, manipulate them. Of course, Galaxy is also an alternative, but command line will make a process much smoother in order to manipulate them, and just in case I have a huge workflow, I can directly write a code and give all of them together in one go. So Galaxy does take a lot of time, and in case there is any web page error or server error, then uploading the data again and carrying forward takes a lot of time here. For tomorrow's class, what we'll be doing is we'll be going forward with the alignment and variant calling, and how do we use the annotation. But before that, again, I will go through all the data downloading and how do we go with quality check analysis, and then I'll proceed with alignment again. I hope you're able to download the data, raw data, which I'll provide the link again. Uh, yes. So for parameters, you can mention if you need the Galaxy. Just a second, I'll just access and I'll let you know. See here, as it mentions, all of these are optional. Adapter list in Galaxy, you'll have to Mr. Rohit, sure. I'll just finish the Galaxy thing and I'll give it to you again. Brief the SRA thing again. Ms. Aditi, to answer your question, once we select the raw data, the contaminant list, adapter list, everything in Galaxy, we need to give it a tab separated file, a text file, which is not really necessary because you may or may not have the data of contaminants and adapters used. And if you have, then well and good, you can make a tab separated file for all of the reads and give the adapters there. Otherwise, as it mentions, they're all optional. And here you can disable grouping of bases for each greater than 50. You can always have it no because that's a default. The optional Kmer again, 7 is the ideal Kmer, but you can also select if you want a higher range Kmers like 10 for your study. But I have gone forward with default job resource parameters in Galaxy, and even in Terminal also I usually proceed. You can usually proceed with just the default parameters. You can once have a go through with the result it provides, and accordingly you can rerun fast QC with the new parameters you always want. Like you can click for you can choose for duplication, no duplication. You can use for KMers and everything. As I had mentioned in the terminal, it always gives us a lot of options. Sorry about that. So, so here it has the KMer option. The default KMer option is 7, so you can leave it as it is, or if you want to increase or decrease the KMer size, it's up to your choice. Adapters also, if you know the adapters, you can always included, but FastQC is programmed such a way that it definitely takes in consideration Illumina adapters, which are standard adapters. So we need not worry. We can always mention the threads we want. Contaminants also, if we know, then we can specify a file, tab separated file, and we can give it over there. Out directory means it will give us all the results in a different directory or different folder than the current one. Since I did not mention, it is given me in the same folder, all the HTML files for visualization. Uh, Mr. Rohit, how to select KMR? It depends on the study you want. Default for Illumina sequencing analysis is 7. I can get back to you in more detail maybe tomorrow because Mr. Rohit, to download SRA database, you go to GEO website, and then once you open GEO, I hope you can see my slides screen. And once you go to GEO, you type the keyword. For us, it is WGS analysis. So I type WGS, and it gives me a hyperlink, which I click on, and I select any study of interest. I can also go with the mice WGS study, for example, here. Then I have to make sure that the study I'm looking for has performed only on WGS here. Otherwise, I might end up taking RNA sequencing data sets also, which I need not want for WGS analysis. And then I'll go for SRA ID, and I click on SRA ID. It will give me all the list of all the samples that are used in this study. So this page gives me a list of 13 samples, for example. And to download each data individually, I go on each of the site and I click on the run ID, SRR run ID, and then I go for data access. I can also go to FASTA or FASTQ download, but this is just 1.4K basis. So I can directly download this, and it will give me a cloud link. I can directly download this, or I can use wget in terminal and copy paste the link, and it will be downloaded. Uh, you will have to install Sapnita. You will have to install wget command in your system. Any queries? Is everything clear until now? Hello? Any queries, please switch on your audio and ask me. Yeah, you're welcome. Go ahead. Starting part of Linux. Do you mean the downloading part, or Dr. Ahmed? What exactly do you want in Linux? Can you please specify? For Linux, yes. So can just give you an example. So this is how it will be specifying. Uh, Basic command I can give you is to go to the current working folder. You just have to first check which folder exactly you are in. That is pwd is the present working directory command, and then it will show you which working directory you are present in. And to list the files in that directory, you have to use ls. What I can do is I can give you a basic Linux command which I have used in this workshop right after the workshop ends, and maybe you can use it accordingly. It's a very simple operating system, and you can just so you need to know exactly the commands you use for what purposes and some by hearting you. But for fast QC, these are the commands in Linux you have to use. Again, this I will be sharing it with you all once the workshop is done. Uh, it need not be fast QC. It can be in a different folder also. Maybe you can make a folder called result, and you can specify the folder name, and you can copy paste there as well, or you can even do manually. Like for example, if I need it to be in a different directory, I create a different directory called result. I give fast QC all the files. Then I mention O, that is the output directory as mentioned here. O means the, I'm sorry, this is getting stuck. O is the output directory I want, like I needed result directory, and it will just run direct. I'll mention the command as well. Mr. Nawaz, may I know your query? Hello. Any doubts for today's session? Tomorrow again, I shall be going through the data download. Sure, I'll also make a list for the Oxford Nanopore reads, but I have specialized in Illumina sequencing data analysis, but I can also give you a link for the same. I'll be sharing. Uh, yes, you can, after you have downloaded the SRR file, you can convert them to FastQ files and you can run the FastQC just in case you want. Yes, Mr. Nawaz, please let me know. Yes, Mr. Thangam. I'm pretty sure you have the file called SRR and the ID name. So once you have that file, you can use this command fastq dump space split files and your SRR file. In case it shows fastq dump command not found, you will have to install it in Linux. SRE toolkit installation. I'll share the commands for running fastqc. Uh, to convert FASTA GZ file to FASTA QVZ. Fast. Yes. So you have a file in FASTA.gz and you need to convert into FastQ file. Am I right? Okay. Yeah, I will share the command for this as well. Conversion of FASTA file to FastQ file, but that will actually lead you to false FastQ quality scores. You will actually have a FastQ file itself. Once you are sequencing, it's better to have a FastQ file because they have quality scores. I can show an example of FastQ file and why we use FastQ files for the analysis. One second. Uh, yeah, I'll just let you know 1 second. Once this file is downloaded, we need to save in fast QC format. We need to save in fast Q format. Usually there are two files. One is pasta file and other is .fastq file. For analysis, we use .fastq file because it provides quality scores for each reads. FASTA file is a little bit of a problem because it cannot provide FASTQ file quality scores, which is required for most of the analysis. Otherwise, we'll have a very duplicated or false quality scores, which may be misleading for our analysis. So this is an example of how a FASTQ file looks. This is the SRR ID file name. This is the header line for the sequencing, Illumina sequencing. It also gives us the length of the sequence. This is the basic NGS sequencing file that is of 150 length. And then this is the sequence file. For the line I've underlined, this highlighted as the sequence. Then the line, the last line that is below the plus sign, this gives us the quality scores. These are ASCII quality scores. These are the quality reads for each of the corresponding nucleotide. Like for C, it is F. For A, it is F. So this is how each of the tool we use for analysis interprets the quality for each of the read here. And we need to have FASTQ files for perfect non-misleading files. Otherwise, we might have qualities, poor quality reads, and we'll be using it for an analysis, and we'll end up with misleading data in the future. Any more questions? Any doubts?
Miss Ishari, today's session is over.
Yes, sir. For now, if they don't have any more queries, I can
If you have any queries,
conclude
you
the
can
session.
ask so that you can utilize this time. Otherwise, tomorrow what you will be discussing?
Yes, tomorrow I will once again go through how do we download the data and how do we install the tools and how do we use Galaxy Workbench for an analysis, and I'll proceed with the further downstreaming analysis, alignment, variant calling, and annotation of the variants.
Okay, okay. Using Galaxy, is it?
Yes, Galaxy, and if those who have Linux also can follow the same
Okay.
Linux terminal. I'll be showing both.
I think some query is there in the chat box.
Yes. If in case any command is not found, you can just uh, install it using uh, sudo or I have saved the file. No, no, Miss Arya, you cannot directly save the file as manually. You'll have to convert the file using fastq dump, which is a tool. Yes, Mr. Pramod, tomorrow I will also be covering command-based analysis and Galaxy, both of them for any data of your choice. If your system is compatible with high data, also is fine with me. Which package is down? In case cut adapter is not downloading, you can also download using Trimomatic tool or BB tools. I will give a list of tools you can download either of them. Sudo, you can just, yes, I'll give a command for example. You can just use sudo and whatever the command like apt get install, whatever installation tool you want, like for example fastqc. So sudo we can just have to use in the initial of the command. There is a limit, but I am not sure how much is the limit. But I will also be showing in command line. But It's up to your choice. You can also use 4 to 6 samples, for example, in the workshop, because I don't want the system to get stuck in between, because alignment tools take a lot of memory and space, and Galaxy as well will take a lot of time in aligning. So it would be very helpful if you all could manage downloading the data today itself and converting them into read one and read two files, so that tomorrow I'll also Linux and troubleshooting. Yes, I shall share a general Linux workbook. Uh, yeah, either of them is fine, Mr. Pramod. But for WGS data analysis, make sure to download sample file and a test file also, like a norm. Like for example, one will be normal data set and the other will be tumor data set or a disease data set, so that we can understand the variance and we can compare them. Uh, Today, after the totally, there'll be 4 FASTQ files, 2 for sample and 2 for test. Each sample has 2 FASTQ files, read one and read two, because this is a paired and sequencing files. I'll also share links in case you need to read through the theory as to how do we interpret read one, read two files, and etc. for your extra information. This is currently I've just shown the fast QC analysis, that is the pre-processing and quality. This is the same for both reference based and de novo. Tomorrow I will be showing alignment onwards. So for reference based and de novo based, from the alignment the pattern changes. For whole exome sequencing, there might be minor change, but pipeline will usually be the same until alignment. Sure. Sudo command is sudo and whatever command, for example, apt-get and the command install, whatever command you are looking for. In case, yes, we'll be using GATK for variant calling. We can also, if GATK is not installable, I'll give you links for Freebase and SAM tools, which are also variant calling tools. I'll give a list of all the tools, and you all can download based on your choice. Uh, you will have to update Mr. Thangam. Maybe that is the reason why sudo is not recognized. You can also copy paste the error. In case in Linux any error throws up, you can just copy paste it in Google and you'll get the result accordingly. Please check the message box for tomorrow's session. I will. It will be easier if you could download the data, convert it into FastQ, and finish with the FastQC analysis. You can take 2 3 samples of your choice, but make sure to have a normal sample and a test sample. Is everything clear?
Everything clear? No more questions?
Normal and test in the sense, you if you in the studies, if you go, you will see the suppose the studies have conducted in a tumor based analysis, then you can download two normal data samples and two test samples or tumor samples. So in the studies you will have to check accordingly. For example, mm, like in this example, if you can see my screen, this is a tumor tissue. It will also have I'll just show it to you. So, like in this study, they have taken nine samples, in which the first two are normal samples, that is from a normal human without any patient samples, and the rest, all the other seven samples are patient samples. That is test samples. So, I'll take of my choice. I'll take two normals and two test samples, or four test samples, anything as my choice, because this is required for comparing. I will also share the geo data link so that you all can also download the samples and keep. This is minimum of 4 GB data. Each data set is of minimum 4 GB. Yes, to convert the SRR file to FASTQ file, you have to use FASTQ dump, which is the tool you'll need to install in your Linux environment. Miss Arya. I'll also share a link how to convert it into FASTQ file soon. Is it clear until now? I have given the link to download the samples. Maybe this will help. Uh, any further queries? Yes, you need Linux environment for converting into FASTQ file. I'll also check for alternative if you can convert a file to FASTQ file, or if you can obtain a data directly as FASTQ files, you can also proceed with it. In case you all can't convert it, I'll share a drive link which have FASTQ files. I'll upload four samples in it. You can directly download the FASTQ files from the drive link for tomorrow's analysis. I'll upload and share it by tonight. Sure, sure. I will share a Google Drive link with the samples. But kindly do not open the FASTQ files in your Windows system to read it in the text file, because they are very huge files and you will have problem in operating it. Yeah, if that is to Galaxy also, if that is possible, please go ahead with it. Or I'll also upload the raw data tomorrow. Because FASTQ dump is not there in Galaxy. You need a Nasare toolkit, which I'll just check. Miss Princey, may I know your query? Miss Princey, please tell me. You're welcome, Tangam. Okay, Mr. Abraham.
So we can wind up.
Yes, sir, Dr. Noble.
Any more query? I'll just wait for. We will just wait for 1 minute. Any query? Please ask. Otherwise, we will wind up, and tomorrow we will continue with this session.
Yes,
Hope you are benefited by today's session. Tomorrow, I think you will be.
tomorrow again I'll walk you all through how to download the data and everything, and I'll upload the raw data sets in Google Drive today. Hopefully, you can download it. But please do not open the files in text editors.
You will
It'll
get the.
crash your system. Yes.
You will get the recordings today itself. You just go through that also before attending, so that you will get an idea what all things we have done. So okay,
Yes.
okay.
Yes.
Thank you,
Thank
everyone.
you,
Thank you.
everyone.
Thank
Yes.
you.
Thank you, Dr. Noble.